Our Alzheimer's project focuses on how gene therapy can lead to the prevention and treatment of Alzheimer's.
Alzheimer's is a progressive disease and a form of dementia that alters a person's memory, behavior, and thought. Symptoms include forgetfulness, difficulty learning, changes in personality, confusion, and learning disabilities. Causes include genetic, environmental, and lifestyle factors. Diagnosing the disease is difficult because a specific test does not exist, but physicians can determine if a patient has the disease through medical history, mood tests, neurological exams, and other tests to eliminate dementia-like symptoms. Recently, researchers have studied a virus that is able to intervene with the disease in order to prevent symptoms. As we see in the first picture, the first step is to identify gene variants associated with the disease, demonstrate functional links between these gene variants and pathology, and then we can move on to prediction and prevention, which would lower the occurrence of Alzheimer's in the general population.
The two most significant differences between a healthy brain and a brain affected by Alzheimer's are neurofibrillary tangles and amyloid plaques. Studies conducted are trying to focus on how to prevent and treat these plaques and tangles. Researchers have prevented the development of Alzheimer's disease in mice by using a modified virus called lentivirus. The lentivirus has also been used in other disease research such as Parkinson's. The virus works by delivering a specific gene to brain cells. The gene is called PGC1 alpha, and it prevents the formation and accumulation of the protein amyloid beta peptide. This protein is the #1 cause of amyloid plaques. The study also showed the mice that had been treated early and directly after symptoms had been shown developed significantly less plaque and performed better in memory tests than the mice that had been treated later. Another treatment scientists have recently found are treatments using ASOs. ASOs are antisense oligonucleotides, and they work like modified DNA-like molecules. They recognize and cleave specific RNA sequences in order to decrease the production and the existing levels of tau. By reducing levels of tau and preventing its formation, stable microtubules will be able to flourish without tangles.
Another treatment that scientists have found are NGF, or nerve growth factors, which maintain neuron health. They encourage the survival and proliferation of subtypes of sensory neurons. The acetylcholine-producing cells that live in the basal forebrain area disappear during Alzheimer's. Scientists have linked those cells to an importance in brain cognitive development and function. Cell death and neuron degradation are obviously very important components of Alzheimer's, and NGF works to minimize those both. A small clinical trial of about 8 patients was used to prove the effectiveness of NGF, and the beneficial effects were found decades later in cognitive development and function, as well as memory tests and general brain function.
Ultimately, genetic research opens doors to treatments and preventions for Alzheimer's disease as technology advances. By interfering with tau tangles and amyloid beta plaques, cell communication can thrive and lessen symptoms and may even eradicate the disease altogether. Here's our bibliography, and we thank you for watching.